Welcome, Brookings Bio students. This is Mrs. Rydell, and today we're going to look a little more at some of the genetic information that we've learned about since Mendel's time. When Mendel looked at his pea plants and made his crosses, peas are a pretty simple organism to do genetic crosses with. The traits that he was looking at only had two choices for their alleles: tall or short, wrinkly round, yellow green, purple flowers, white flowers, and it was pretty easy to see the patterns when you made those crosses. But since that time, we've learned a lot more about genes and how they work, and genes are a lot more complicated than Mendel first thought. So we're going to look a little more closely at some of the other things that can affect how genes work. One of the things that we know now is that environment plays a role. Environment influences the expression of genes, how the genes actually show. And we talk about nature versus nurture. Nature is the genes that you receive from your parents, but they're influenced or impacted by the things that you experience as you go through life. Nurture is how environment plays a role in how genes are expressed. And so we know now that genes kind of provide the plan for development, but how that plan unfolds and what the ultimate phenotype of an organism is depends on environmental conditions. You are the way you are not just because of the genes you inherited, but by environment, how environment impacts those genes as you grow. Example. Hydrangeas, some of you may have seen hydrangea bushes growing in a yard. Hydrangea flower color is influenced by the pH of the soil, and you can actually change the colors of your hydrangea bushes by adding things to your soil to change the pH. They can be pink, they can be purple, they can be blue, depending on the pH of the soil. Another example of how nature, environment influences the expression of genes. If you have ever seen a Siamese cat or a Himalayan rabbit, you know that they have a very distinct pattern to their fur, dark in some areas, light in other areas. And the allele that controls the pigment production, the color production in their fur, only works at lower temperatures. So when places in their body, their main body, where they have higher temperature, that pigment is not turned on, and the bunny or the cat looks lighter colored. And in places where they're cooler, out on their extremities, their legs, their feet, their ears, their tails, that gene, where the temperature is cooler, that gene is going to be expressed, and we see places on the cat or the bunny rabbit that are darker in areas where they're cooler and lighter in areas where their body temperature is warmer. And it's the same gene, it just acts different at different temperatures. Some traits have more than two allele choices. When Mendel looked at his peas, all the crosses that he made only had two choices for their alleles: tall or short, wrinkly, round. But many genes are controlled by what we call multiple alleles. There's more than two choices. Each person, each organism only gets two choices. You have two homologous chromosomes, but there are more than two possible allele choices for that trait. Example in humans is blood type. And in blood typing, we have three allele choices instead of two: A, B, and O. And so now that complicates things a little bit more. Some traits are determined by more than one gene. The traits that Mendel looked at, flower color, seed shape, height of plant, they were only controlled by one gene. But many traits, most traits in humans, are what we call polygenic. They're controlled by more than one gene. Think human height, intelligence, skin color. When we see a huge variety of different allele choices, it's probably telling you that there's more than one gene involved in that trait. Polygenic. Remember we had polymer? Poly means many. Polygon in math. Polygenic trait is controlled by more than one gene. if you have a trait that's determined by more than one gene, one clue is that you have lots of in-between phenotypes. If you think about it, we don't just have smart people and dumb people. Two choices. You're either smart or you're dumb. There are lots and lots of in-betweens, a whole range of intelligences. Think about other human traits. Height, for example. Height is, we don't just have tall people and short people, like when Mendel looked at his pea plants. We have lots of in-betweens. If I stood everybody in this room up and lined you up by height, we'd have a whole range of heights. Skin color is another one. Multiple genes involved in controlling skin color, height, intelligence in people. And so that would be those are examples of polygenic traits. The other thing when Mendel looked at his pea plants is all the genes acted in a certain way, that dominant recessive interaction. If the dominant gene was present, the recessive gene didn't show, and that's not true for all traits. So we're going to look at some other kinds of dominance besides Mendel's complete dominance, where the dominant one always totally covered or hid the recessive. The other kinds of dominance we're going to look at are incomplete dominance and what we call co-dominance, cooperating, co-dominance. So let's look a little bit closer at those patterns. When Mendel looked at his pea plants, they showed complete dominance for the traits that he experimented with. The dominant allele always masked or hid the recessive one. If the dominant allele was present, the recessive didn't show. So when he crossed, for example, if he crossed a red flower with a white flower, all the first generation, the F1s, all had red flowers. And then if he crossed those organisms with each other, in the next generation, the F2s, remember that little recessive allele came back again, always in that 3-to-1 pattern. Recessive returns in the F2 generation. That's the pattern that we see if a gene is acting completely dominant. In incomplete dominance, though, we get a pattern that's different than what we expect. We don't see that expected 3-to-1 ratio in the F2 generation, and we don't see the pattern that we'd expect all dominant phenotype showing in the F1 generation. Organisms that are heterozygous, they have one dominant and one recessive allele. They show a blended or in-between phenotype. So for example, a gene that is incompletely dominant, if you crossed red flowers and white flowers, you don't get red flowers, the dominant allele. This is this is incomplete dominance now. Red and white, you get pink flowers. Pink flowers. If you're heterozygous, you get the blended or in-between phenotype. And if you cross those pink flowers with each other, you don't see a 3-to-1 ratio. You see a 1 to 2-to-1 ratio. You see some red flowers, you see some pink flowers, you see some white flowers. The pattern is different, and that's how you can tell a gene is an incompletely dominant inherited trait. It shows a different pattern than that 3-to-1 ratio. An example of codominance is where both traits show at the same time now, is what we see in horses, horse color. Codominant traits an example of an organism that shows this incomplete dominant flower color are flowers called 4 o'clocks. 4 o'clocks are flowers that bloom in the late afternoon. That's where their name comes from. But they show incomplete dominance in their flower color alleles. So if you cross red 4 o'clocks with white 4 o'clocks, you don't get red offspring. You get pink flowers. And if you cross those pink flowered organisms, you get some red flowers, you get some pink flowers, you get some white flowers in the F2 generation. They're showing incomplete dominance. The other kind of dominance, excuse me, the other kind of dominance is what we call co-dominance. Both traits are expressed at the same time. It's not a blend like we saw in incomplete dominance. They both show together. An example of this is roan color in horses. If you look from a distance at a roan horse, they kind of have a light tannish color. From a distance, they look kind of a tannish brown. But if you get up close and really look at their hair, what you see is they have both reddish color hair and white hair together. It's not all one color of hair. You have both kinds of hair growing on the horse, and they grow side by side. So when you stand at a distance, it kind of looks like a one color. But this is not a blended horse. This is not a pink horse. Red crossed with white, you get a pink horse. You get both red and white showing together side by side. That's co-dominance. They're both cooperating to give you the color. Another example of co-dominance is what we see in human blood types. Both traits being expressed. People who have an A allele for blood type and a B allele for blood type have what we call AB blood. They show both of those characteristics, the A sugar and the B sugar on their surface. I'm going to talk a little bit about blood type. Remember back in our molecule chapter, we learned about molecules that attach to the surface. They have sugars, and they're made out of proteins with sugars attached, and they stick on the surface of cells just like little trees, and the sugars help in recognizing self. Who remembers what those are called? Remember glycoproteins. Glycoproteins are involved in cell recognition. Are you like me or are you different? And really, this is a good example of a multiple allele trait. Remember our more than two choices? Remember, not just tall or short, wrinkly or round. We had 3 choices for blood type alleles. The A allele, the B allele, and the O allele. And really, what's happening here is these are proteins that are coded for by the A gene or the B gene or the O gene, and they are put on the surface of cells. So how blood typing works is if you have an A allele, what it tells the cell to do is make a certain kind of glycoprotein and put it on the surface of the cell. It sticks out on the surface, those little sugar molecules. And we don't have to really be worried about what the actual shape of the sugar is, but a certain kind of sugar is put on the surface of cells that have that A allele. If you get the B allele, it makes a different glycoprotein. The sugars that are on the surface of B cells, persons that have B allele, they have a different kind of glycoprotein, a different sugar pattern on their surface. And we would say they have the B allele gives you B glycoproteins on the cell. People who have the O allele is actually an A allele with a mutation. It's lost some of the gene code, and so it doesn't really work anymore. An O allele is a broken A gene. And what it tells the cell to do is don't put anything out there. It doesn't make the glycoprotein that it normally would. And so the surface doesn't have either A or B on the outside. It does not have an O glycoprotein. It's not telling it to make an O glycoprotein. It's saying don't make anything because the gene that makes A and B doesn't work. Does it mean there's nothing on the surface? No, there's lots of other things out there. It just doesn't have an A or B allele choice. When you do blood typing or match with for organ transplant, there's multiple alleles, probably 10 different kinds of glycoproteins that they look at and match to make sure that you're a good donor match. But we're just looking at the A-B choices for a certain kind of glycoprotein. And people who have O blood type don't have either A or B glycoproteins on the surface of their cells. If you have both alleles, remember people have 2 of every chromosome, so if dad gives you an A and mom gives you a B, you're going to have both choices. And remember, this is a codominant trait. That means both alleles are going to show. So the A gene tells the cell put A's out there, and the B gene, B allele, tells the cell to put B glycoproteins out there, and the cell looks like this. It has both kinds of glycoproteins. They both show together. Neither one is dominant over the other. They're co-dominant. Both show. So here's how this blood type thing works. If you have A, both allele choices, remember homologous chromosomes, you have two alleles that are both A's, they're going to tell the cell to put A glycoproteins on the surface, and the phenotype is going to be A blood. If you have an A allele choice and an O allele choice, A is dominant to O. O acts as a recessive gene because remember, it's broken. It doesn't really work. It doesn't do anything. So anything that's mixed with O, that's the part that's going to show. So O acts recessive to the other allele choices. So if you have A glycoproteins on your surface and O says do nothing, this person is also going to look like they have A-type blood. They're going to have A glycoproteins on the surface of their cells, even though that little O allele is present. It's kind of like when you have big T, little t, and the little t recessive allele is there, but big T is what we see. The plant looks tall. And same with O being recessive in blood types. If you have an A and an O allele choice, A is going to show and O is hiding. If you get 2 B alleles, you're going to have what we call B-type blood. And if you have a BO choice, remember O acts recessive. It's broken. It doesn't work. So we're going to also have B-type blood. The B allele is going to tell the cell put B glycoproteins on the surface. And O is hiding. We don't see. What if you get 2 O alleles though? Remember, neither one of them are working. That doesn't make a glycoprotein. So we would say you have O-type blood. Do you have other glycoproteins on your surface? Yes, and we're going to talk about another one in just a minute. But for the AB blood group, that person we say has O-type blood. If you get both alleles, A and B, we say you have AB blood. AB blood, because both alleles are showing. The A glycoprotein is there and the B glycoprotein is there. So let's look how this thing works and how cells use these glycoproteins to recognize self. So we're going to stand up our people here and you kind of have a blow up of their cells so you can see what kind of glycoproteins they have on their surface. And our donor blood now is going to be up at the top. We're going to give a donation, a blood transfusion to these people and see what happens. So if you have donor blood that has A's on the surface and you try and give it to a person who is A-type blood who also has A's on their surface, okay, both of these people are going to see that A blood and not see anything on the surface that they recognize as that's different than me. The A person has A's already on their surface. They're going to see A blood as that's like me. They have A's just like I do. The AB person, even though there's not any B's on this donor blood, they have A's just like this person has A's. And they're going to say, hey, you got A's, I got A's, you're like me, you're not different. But if you try and give A blood to a B person, that person's body is going to recognize that those A antigens, we call them antigens, those A glycoproteins are different from what this person has on their cells. They're going to see that as a difference. And likewise, an O person is going to see those A glycoproteins as that's different from what I have. That's different from me. And their body is going to attack those cells and cause clotting, and we get a reaction to the donated blood. What if my donor blood is B-type blood? B blood can be given to another B person. They're going to see that as, hey, you got the same thing I have. Also to an AB person, because AB has Bs, they're not going to see those Bs as different or something different than what they have. They have Bs, donor blood has Bs. You can give B blood to an AB person. They're going to see that as, that's like me. If you try and give B blood to an A person, that person is going to recognize that is very different. Bs are different than As. And likewise, an O person is going to see those glycoproteins on B blood and say, that is different. You have something different on your cells than I have. And so an a B donor given to A person or a B donor given to B blood, those people, their immune system is going to recognize as that's different and attack those cells. You're going to see a reaction. What if I give O blood to these people? O is what we call the universal donor. It doesn't have A's or B's. There's nothing on the surface of that cell that an A person is going to see as different. It's kind of invisible. Remember, the O gene is broken, so it doesn't put A's or B's on its surface. And so the A person is not going to see B's. They don't see that as any different. The B person is not going to see any A's out there. It's just going to see it's invisible. It's kind of like an invisibility cloak. It's hiding. The AB person also is not going to see anything on the surface that recognizes that's different than me. And the O person, oh, hey, you're like me, right? All of those people can receive O blood. O is what we call the universal donor. In a pinch, if you're at the hospital and there's an emergency and they run out of blood, the blood to have on hand is O type blood, because O is in a pinch can give to everyone. There's nothing on the surface of that O cell that those people are going to recognize as that's not me. How about an AB person though? An AB person, I can give AB blood to another AB person, but if I try and give AB blood to everyone, we're going to have some problems. And another AB is going to see that as, hey, you got what I got. But an A person, even though these have A's, it's going to recognize those B's on the surface as that is very different. A B person is going to recognize the A's on the surface as different. And the O person is going to say, oh my gosh, that is so different from me. Giving AB blood only works if you give it to another AB person. AB donor is going to react with A, B, and O. And so we call the AB person the universal receiver, right? I can give O to an AB person. They don't see that as different. I can give A to an AB person. I can give AB blood to an AB person. I can give B blood to an AB person. AB can receive every kind of blood type. We call the AB person a universal recipient. They can receive all different kinds of blood. Just to give you an idea of frequencies, the most common kind of blood to have is actually O blood. AB is the rarest form. Only about 4% of the people in the United States have AB blood. And you can kind of see, I don't know if you know what your blood type is, but you can kind of take a peek at the frequencies here. The ABO blood group system is not the only blood typing system that you have. When we do a transplant or a transfusion, we look at other kinds of glycoproteins on the surface. There's about 10 different, we call them HLA matches that they're looking for. And we're going to look at one other blood group system that's important, probably if you're going to have kids down the road. And that's the RH system. The RH stands for rhesus. It was originally, this glycoprotein was originally found in rhesus monkeys, and that's where the RH name comes from. And so if you have the allele for RH, what it tells your cell to do is make a certain kind of glycoprotein, just like we did it with A and B. It's going to make a certain kind of glycoprotein and put it on the surface of all of your cells. It's different than the AB glycoprotein. It's another kind of glycoprotein. And so it puts it on the surface of the cells, just like the we saw in the A and B blood group alleles. And so, for example, if you are a person who is Rh positive, your blood type, when we talk about blood type, is usually say that A or B or O or AB, and then either positive or negative. So I'm O negative. You may be A positive, and that little positive or negative is talking about whether you have the Rh allele or not, the Rh glycoprotein on your cells. So if a person is for example, A positive is going to have both A glycoproteins and those Rh glycoproteins on the surface. If I'm B positive, I'm going to have B glycoproteins and the Rh glycoproteins, both on the surface of my cells. This is a different gene than the ABO gene. If you have what we call Rh negative, it kind of acts like O. It doesn't put any glycoproteins on the surface. So you might still have A's or B's on the surface, but you're not going to have the Rh glycoprotein on the surface. Where this becomes an issue has to do with pregnancy. And this doesn't cause any problems as long as the mom and the baby are both positive. If mom is Rh positive and baby is Rh positive, there's not a problem, because mom isn't going to see anything on the surface of those baby cells that she's going to recognize as that's different and not supposed to be there. Remember, baby is growing inside the mom, kind of like a foreign invader. If the mom is positive and the baby is negative, we also don't have issues, because mom has Rh glycoproteins on her surface and baby does not. So mom is not going to see anything on the surface of those baby cells that she recognizes as that's not supposed to be here. The issue comes where the mom is negative and the baby is positive for Rh. In that case now, the baby growing inside the mom has glycoproteins on the surface of its cells that mom's body is going to see as that's not supposed to be there, that's different. Baby grows inside, protected by an amniotic sac and attached through a placenta to get its food, and there's not a lot of mixing of the mom and baby's blood. Mom and baby blood doesn't actually mix and mom's blood go into the baby when the baby's growing inside. We attach through a placenta, that's where your belly button came from, and the mom and baby blood vessels get very close so they can exchange oxygen and food and nutrients and collect the garbage from the baby, but they don't actually mix the blood of the mom and baby. So baby grows safe inside the mom, but the problem comes when the baby is born, because when the baby separates from the mom, there's some bleeding, and some of those baby cells can get into the mom's bloodstream. And now the mom is going to see those baby cells in her body as that's not supposed to be there. It doesn't really harm the first baby because that baby's been born. But her immune system now remembers those cells. One of the things your immune system does when it's exposed to something that's foreign is it has a memory. It remembers those glycoproteins it saw. So the next time that it sees those again, it's ready to attack and get going right away. And that's the reason why usually don't get measles more than once or chickenpox more than once. Once you're exposed to a foreign kind of disease, foreign kind of glycoproteins, your body remembers those. So even if a few get in later on, they're going to get pounced on right away and destroyed, and they don't get a chance to get started and make you sick again. So the problem comes with the next pregnancy. If the baby is positive, again, a second baby, and the mom is negative, now her immune system is triggered and ready to recognize those baby cells, and her antibodies can actually cross into the placenta and get in and attack the baby that's growing inside of her. It causes a problem called erythroblastosis fetalis, and the mom can actually, her body, her immune system can actually attack and harm the baby that's growing inside her. So how do we fix this? When the first baby is born, mom is given a shot. It's called RhoGAM for the RhoGAM, and it prevents her immune system from getting that memory to recognize those baby cells. So the next pregnancy is not going to be seen as you're not supposed to be here. One of the other things we need to talk about to do Punnett squares has to do with determining sex. And you have two chromosomes that determine the sex of the, whether you're a boy or a girl. We call them sex chromosomes. All the other chromosomes you have in your cells, remember you have 46 chromosomes in all your body cells. All the other chromosomes are called autosomes. And since we have 46 chromosomes, then humans have two sex chromosomes. We call them X and Y. Kind of like using letters to stand for something in math. And then that means you have 44 autosomes. 44 autosomes. And the presence of those X and Y chromosomes now are what's going to determine whether you are a male or not. The default is XX. If you get an X from your mom and an X from your dad, you are going to be female. Everyone starts female. Boys in this room, you started as girls. The basic anatomy is everybody starts female. If you inherit the Y chromosome, though, there are genes on that Y chromosome that turn on and start making you into a boy when you are a very young embryo. And if that Y is not present, then you're going to get a female baby, and X-Y combination makes you a male baby. So let's Punnett square with those XY chromosomes. We're going to cross a mom. Remember, mom has two X's. We split them up to make gametes, X or X. Mom can give X to the baby. Dad can give either X or Y. Those are his allele choices. So if we put them together, mom gives X, dad gives X, we get girls. If mom gives X and dad gives Y, we get boys. So think about that for a minute. Because mom only has X chromosomes to give, doesn't matter what sex you are, you're going to get an X chromosome from mom. What really determines your sex is what choice dad gives you. If dad gives you an X, you're going to be a girl. If dad gives you a Y, you're going to be a boy. So dads are the ones that determine the sex of the baby. A long time ago, there was a king in England who spent a lot of time trying to have a son. In those days, the kingdom passed from fathers to sons. Dad wanted a male heir to inherit the throne. And this king spent a lot of time going through wives. He either divorced them or had their heads chopped off. And he would get a new wife and try for a son, and spend a lot of time going through a lot of wives. And the irony is that it wasn't their fault. The dad is the one, the king was the one who was determining the sex of the baby. We have one last thing to talk about when it has to do with how genes are inherited. And some genes are inherited on autosomes. Some genes are carried on those sex chromosomes. There's a mutation called HEMOPHILIA that codes for genes that are blood clotting proteins, and it's carried on the X chromosome. So if this mutation happens, that means a person who has that allele is not going to be able to make the blood clotting proteins they need. They're missing. So if they get injured, normally what would happen when you have an injury is those blood clotting proteins would clot the blood, clog up the blood, and makes a little scab, and you stop bleeding and the repair starts. But people who have this mutation on their X chromosome can't make those clotting factors. They don't stop bleeding. And they can actually bleed to death from a very minor, just like a paper cut or a little bump or bruise. And that gene for hemophilia is carried on the X chromosome. So if we have a gene that's on a sex chromosome, we're going to see a little different pattern. Hemophilia, people who have this need to get injections, and if they get a bleeding incident, they go to the hospital and they receive those clotting factors through an injection to help them to stop clotting, stop the bleeding and clot their blood. And think about this now. because it's on the X chromosome, it's more common in males. Remember, females have 2 X chromosomes. In order for a female to show this trait, she's got to get 2 of the mutations on both of her chromosomes. Males only have 1 X chromosome. So if they get a mutation on that 1 chromosome, they're going to show the trait. They don't have a second X, a backup X, that can make those clotting factors. Females have 2 chromosomes. So if they get a mutation on 1, they still have a chromosome that has the normal gene that can make the clotting factors for them. 1 in 10,000 babies, male babies, has hemophilia in the United States. Another example of a trait that is carried on the X chromosome is color blindness. The proteins that make the receptors in your eyes that receive the information about color, the gene for those is carried on the X chromosome. Actually, there's several genes, and we can have different kinds of color blindness. The red-green, problem determining red from green. If you look at this, you can see these little dots here are a different color than the ones on the outside. People who have color blindness can't distinguish those 2 colors. They don't see a number here in the middle. And color blindness is an X-linked trait. It's more common in males. 1 in 10 males are colorblind. 1 in 100 females is a very common phenotype that we see. So if you think about how this works now, remember, males get 1 X chromosome and 1 Y. And notice X is a lot bigger than the Y chromosome. This part matches, but Y is actually missing some of its genes up here. But they match as homologous partners. And so if you have a defective gene that's carried on the X chromosome, the Y chromosome doesn't have a normal match to make up for that problem. If you have the normal allele in males, a normal X chromosome in a Y, they're not going to show the trait. But if you get the recessive, it acts like a recessive trait. If you get the gene, it's going to show. They don't have a backup. They don't have one to make the normal protein. So females, because they have 2 X chromosomes, they can have 1 normal gene and 1 mutation, 1 defective gene, like hemophilia or color blindness. Because they have a normal gene that works, this one's not going to show. They're able to make the things that they need to not show the trait. In order for females to show a recessive trait that's linked on an X chromosome, they have to have both of their homologous chromosomes have to show the defect, have to have the mutation in order for it to show. So X-linked traits more common in males. Males only have 1 X chromosome, so it's kind of like flying without a co-pilot. There's no backup chromosome to make up for that missing or damaged gene. The other thing we need to talk a little bit about when we talk about human genetics is the idea of heterozygous people having both choices. If I cross a person who is heterozygous for a trait, for example, Tay-Sachs is a trait that's carried on autosome. It's a regular chromosome. And so if the dad has both a normal gene and a Tay-Sachs gene and happens to meet a mom who also has normal with a Tay-Sachs gene, they don't show the trait. They don't see the trait in the parents. But if those 2 little alleles get together, they can have a baby that shows the genetic sickness, has Tay-Sachs. It's a pretty terrible disease. Babies become mentally retarded, and they go blind, and they die at a very young age. And it can show up in families where we don't really, it hasn't shown up before because this person has the allele but doesn't show it. And we call those people what we call carriers. They're carrying that recessive allele. So even though they don't show it themselves, they are carrying the allele, just like big T, little t, the recessive alleles there, but we don't see it. And that same thing can happen in humans. And we call those people, they're heterozygous. They don't show the genetic trait themselves, but they're carrying the allele and they can pass it on to their children. We call them carriers. It's one of our vocab words that's not in the glossary. You might want to look at this definition and get it down on your vocab list. We're going to stop right there. We're going to do some punnets with these kinds of, looking at this kind of genetic crosses, and stay tuned for more on genes.